So we're going into the last part of chapter 5. We've done carbohydrates, we've done lipids, we've done proteins, and now we're going to do nucleic acids, which you are familiar with if only from their initials, Na. There's two major types of nucleic acids. There's DNA, deoxyribonucleic acid, and RNA ribonucleic acid. You don't need to know the deoxy and ribo stuff. It'd be nice if you could recognize it. Okay, I guess I annoyed the camera by stepping into the field, as I try to make it so this is easier to see than me. And at this point, we're far enough in that I hope you're doing this in a way that's being efficient for you, that you're actually watching the videos and taking notes rather than trying to hunt through stuff in order to find answers to the exams when you've got a fairly short period of time to do it. But you can actually look through your notes, especially if you're using the videos and actually marking on your notes occasionally what timestamp you are at, so that you've got, okay, this is this video, and then somewhere down the notes you go like 10 minutes in, and somewhere down the notes you go like 20 minutes in, and then if you do need to find a spot in a video, you look at your notes and you go, that doesn't make any sense anymore. I'll go look at the book, and the book doesn't really make sense anymore. Maybe I need to go listen to the video again. You can go listen to the video again, and you look, well, it's between the 10 and the 20-minute mark, so I'll scan somewhere in there and look for it. And of course, you can look for what's up on the screen when you're bouncing through the videos. Nucleic acids. These are long strings again of repeating units. That's what a polymer is. Remember, proteins are polymers, starches are polymers. These are long strings of what are called nucleotides. In living things, there's really only 5 kinds. There's a lot more kinds than that, but you don't tend to find them in living things. And these nucleotides are sometimes very confusingly called nucleic acid bases. Remember way back when with acids and bases, you go, how can it be nucleic acids and how can they be acids and bases at the same time? Well, it all depends on whether they're in the string or popped loose. Popped loose, they act as bases. In the string, they act as acids. We'll use nucleotides. Nucleotides is a less confusing term. So that's what a nucleotide looks like. It's got a phosphate group, it's got a sugar, see the ring with the oxygen bridge, and it's got a nitrogenous base, so it's got nitrogen in it. Oops, started doing it again, pointing with my shadow. It's got nitrogen in it, so a couple of nitrogens. Here's the phosphate group, that helps to attach to things. Here's a sugar group that helps to attach to things. And this is what makes one nucleotide different from another nucleotide. This happens to be thymine, but there turns out that there are bunches of them. I don't expect you to be able to recognize them. This is just me explaining things more than I probably should explain things. But you've got 4 major nucleotides in DNA. You've got adenine, you've got cytosine, you've got guanine, and you've got thymine. Now, DNA is not just a single string, it's 2 strings running parallel, and because of the way that the hydrogen bonds work, it curls like a spiral staircase, where each of the slats on the staircase is one nucleotide attached to the other nucleotide. But they don't all attach to each other, they pair up in a very particular way. And I'll write this down when I get this stuff off the screen. But you can see it here. Adenine and thymine connect to one another. Cytosine and guanine connect to one another. Now you'll notice that these are long, because they actually attach this way, you know. And these are short. That will turn out to make some sense later when I talk about proofreading. That when you're building new DNA, you pull the strands apart, and then you build a new strand on each one. And they're not copies of each other, but if you're building a new strand and you've got an A over here, you gotta put a T there, because that's what connects to the A. If you got a G here, you gotta put a C there. If you got a C there, you gotta put a G there. So you do wind up making a copy of the old strand, just the old strand wasn't a perfect match. It was what they call a complementary match for the first strand. And we'll talk about that later in the semester. And if you inadvertently connect a short one to a short one, then the strands, the parallel strands have a tight spot. If you connect a long one to a long one, then you get a bump. And that's one of the ways that we've got these little molecules that go, oh, we got a dip, back up, we need to, you know, you did this wrong. Or we got a bump, back up, you gotta do this. So, oops, wrong place. I have too many places and too many things. Now here's the deal with these things. You need to remember the initials for sure. And you need to be able to recognize the spelled out names if I ask you questions about them. But I'm not gonna ask you to memorize the names. It's for an in-class exam that makes a difference. I guess for our exams it doesn't really make a difference. Never mind. Incidentally, some of you know that there's a vitamin called thiamine. That's not the same thing. Thiamine and thiamine, thiamine is thiamine. Now, as I've mentioned before, I've never had the best memory for memorizing bits and pieces, right? I'm not an organizer. I tend to be a big picture person. And so I often have come up with tricks to help me remember stuff. I remembered this when I first learned it by putting the things in alphabetical order. So you have A and C and G and T in that order. And then learning that the inside ones connect to each other and the outside ones connect to one another. This also happens to work because when we start to talk about how DNA and RNA work together, RNA has A, C, and G in it, but it doesn't have T. What it does have is uracil, a U. So if you have an A on your DNA strand and you're building RNA across from it, if there's a C, you put a G in the RNA. If there's a G, you put a C in the RNA. If there's a T, you put an A in the RNA. If there's A, you put a U in the RNA. But that'll be something we talk about more in detail later when we talk about how you take this code and you make proteins with the code. Now we've talked about the code before. We have protein-based chemistry, but DNA-based coding. C-O-D-I-N-G, code. How's the code work? We'll deal with this in a lot more detail later. Let me see if I can fit this in here. A protein is a long string of amino acids. Remember, that's the primary structure. It's the order of the amino acids. It's a long sequence. A gene is a really long sequence, but it's much longer than the protein sequence because it's a 3-to-1 ratio. When you're reading a gene, every 3 nucleotides is going to be used to put 1 amino acid on the protein string. And that 3 nucleotide sequence is called a codon, sort of. Is the codons wind up being not really the nucleotide sequence, but something in the middle, but it amounts to the same thing. It's 3 spots that translate to 1 spot. So we have all of these codes for different proteins. We also have a bunch of different codes for the same protein. Is that it's a very, very, very long sequence of nucleotides. And if you start looking around, you'll find this gene code has a spot where the nucleotide is different. This gene code has a spot where the nucleotide is different. And this gene's code has a spot where the nucleotide is different. And this gene code has a spot where the nucleotide is different, and it messes with what the protein can do. All of those variations of the same code for the same protein are called alleles. Sometimes some people pronounce them alleles. And when we get to genetics, that'll turn out to be a big deal. That's how genetic variation happens, is that we all have codes for the same proteins, but we don't necessarily have exactly the same codes for the same proteins. And in some cases, our codes produce a protein that's different in a significant way than what it's supposed to be. More likely than not, it's screwed up. Right? You start messing around with the codes and start messing around with the proteins, you're way more likely to screw them up than to make them better. But occasionally you make them better too. That's part of how evolution works. And of course, in the proteins, we have enzymes. So all of the other molecules that we have that are lipids or are carbohydrates that we make, we have a code for the enzyme that makes this lipid or this carbohydrate. Glycogen. We have enzymes to put those sugars together in your liver and store the sugars as glycogen. So anything that's not a protein, you have a gene that is kind of once removed from that product. Is it, here's the gene, makes the enzyme makes the product. So your genes do control all of your chemistry, even the stuff that isn't proteins in and of itself, because those enzymes do everything. Decent number of years ago, people started to be able to pull the sequences of DNA apart and look at the sequences. And they were starting to figure out how the codons work and how the genes worked. And they got to the point where they could look at a sequence and go, this is a protein code. And then something very surprising happened. When they started to go through the DNA for us or the DNA for a whole bunch of other animals, and it turns out plants too, an awful lot of the DNA wasn't gene codes. It wasn't coding for anything. And of course, being people, for a while, the folks that had discovered this went, we can't figure out what it's doing. Must not be doing anything important. And so they coined the term junk DNA. I'm sure right now they hate that they did that because it makes them look really kind of dumb. And of course, what's since then we've found out is that, yeah, we've got a lot of DNA code that isn't actually a code for our proteins, but it doesn't mean it's not doing anything. It doesn't mean that there's no explanation for it except that it's junk. And I'm going to give you a list of some of the stuff that non-coding DNA does. And I put the non-coding in quotes because even this is a bad term. Because it turns out some of the non-coding stuff is codes. In your little bitty nucleus in your cells, you literally have about this much DNA. That's how long the molecules are, packed into a microscopic space. It's wound and coiled, and the coils are piled on other coils, and they're packed into chromosomes. So there's a tremendous amount of structure in order to fit a lot of molecule into a really tiny space. And a lot of the DNA is involved in interacting with the spooling proteins and the piling proteins and all this other stuff that we'll talk about later. But it's a very important function. You can't pack your DNA right without it. But it's what they call robust. If you mess with it a little, it doesn't tend to really screw up, your genetics on a big basis. And that's what they found when they were discovering this junk DNA, that they could look from one place to another, and in a lot of instances it would be different. And they went, well, I guess being different isn't hurting anything because we can't see anything that it's hurting. So it must not be doing anything. Of course, at the same time, they found stretches of DNA that across a wide swath of different living things, it was identical. Is that all of the mutations over all of the ancestry had not really messed with this. And the question is, what the heck is it doing that it couldn't stand to be messed with at all? Because being, any kind of changes in it tended to kill off the owner of the changes. And some of that is structural, some of that's connected to other things that are just very important for DNA to be able to do. It turns out that a gene code isn't just a gene code. That if this is the stretch of gene code, that there's stuff at the end and a lot of stuff at the beginning, and some stuff that's kind of nearby that are involved in helping to activate and deactivate the genes, what they call epigenetics. Epigenetics has to do with, right now, all of your cells are not using all of your genes at the same time. Right? Your skin cells are just using a handful of genes that a skin cell needs. Your muscle cells are using a handful of genes that a muscle cell needs. And even then, there are genes that you kind of need to turn on when you're building the cells, but then you need to turn off when the cells become functional. Through our lifetimes, we turn on certain abilities and turn off other abilities by activating and deactivating the genes. So we got a lot of processors that allow us to control which genes we're using or not using at any given moment. And that turns out to be a pretty important part of the DNA. Sometimes we'll get a bacterial infection, and the bacteria will wind up integrating their own genetic sequences, which look different from our genetic sequences, into our DNA. And it just, we make it and we pass it on, we make it and pass it on. Most of the time we don't use it. Viruses do the same thing, right? Viruses get their DNA into cells. Sometimes it winds up getting integrated into the actual working DNA of the cell and getting passed on and passed on and passed on. So if you look through our DNA, you'll find sequences that somebody who really knows what the sequences look like will go, well, that's a bacterial sequence. That's a viral sequence. Just recently, they found a couple of bacterial sequences that we do use that have actually been beneficial for us. They help our immune system react to bacteria, weirdly enough. But we've got these foreign codes, so obviously they're codes, but they're not in use. And we have codes that we no longer use. We'll talk about this later, there's a whole bunch of small molecules that we use to help do stuff that are called coenzymes. They help enzymes work. If we need them in our food because we don't make them ourselves, we call those vitamins. Vitamin C is one of those. Now, your typical cat, it still needs that coenzyme. It's a very important part of our chemistry. Hell, the reason we get them from plants is it's an important part of the plant's chemistry too. But a cat doesn't have a regular vitamin C source in its diet. So it has a gene for an enzyme that makes vitamin C for it. Well, it turns out so do we. Except our diet has had so much vitamin C in it for so long, thousands and thousands and thousands of generations, that we stopped really using that code because we never really needed to make our own vitamin C. And over time, mutation has totally kind of made it useless. We couldn't make vitamin C now if we wanted to, because that old code has mutated into an unusable form. But it doesn't matter to us. We got vitamin C in our diet. We don't need to make it. And you'll find that there's a number of other codes that were useful to our distant ancestors, but for a very long time, we weren't using it. And so if it mutated in a bad way, it wouldn't matter, we weren't using it. And over time, they degrade. You can pull them apart and look for homologies, compare the sequence to to the same gene the cats had, and go, oh, we know what that is. That's the for the enzyme to make vitamin C, but this one doesn't work, the cat one works. So old unused codes, and again, still codes. The other type of DNA, or the other type of nucleic acid, sorry, is RNA. RNA, when I learned about it, there were two basic types of RNA you really learned about. There were a few other that we knew about that were in certain structures that helped take the gene and make the protein using the gene. But it was a handful, and we knew what they were. They were all middlemen in the protein making process. Well, in the last 20 years, people have discovered a whole array of little bitty RNAs that are working in the nucleus where our DNA is and are doing all sorts of things. So many, we call messenger RNA little m RNA. We call transfer RNA little t RNA. For a while, they called these new ones little micro RNA. But as they discovered what different ones do, they started to give them all sorts of different designations. And there's so many designations now, I can't keep them straight. I'll pick up an article and it'll have, little NUN RNA. And it's like, are RNA going to this, becoming nuns now? What the heck is this? But it's a really big part of recent discoveries. And of course, how do you make those RNAs? Well, there's stretches of DNA that are used as a code to make those RNAs that didn't look like gene codes, so nobody knew what the heck they were. Didn't know to look for them until they found these weird little RNAs. But RNA turns out to be very much a big, important processor in the nucleic acid world. So, this is my chance to introduce RNA. Later we'll talk about the big ones, the messenger and the transfer RNA. Here's where it gets weird. In some cases, we have stretches of DNA that are in it only for themselves. They act like they were molecular parasites. So they make more of themselves, and they're capable of popping copies out and sticking them other places in our genome, in all of our DNA. For a lot of cases, they're in the non-coding parts, so they're not disrupting too much. But occasionally these parasites get into a code and will mess with what the code is trying to do. There is, oh crap, Huntington's disease. I was drawing a blank on the name of the disease. Huntington's disease, where there's a DNA parasite that is just a codon repeating over and over and over again, that takes the gene code and stretches that middle part of the code out, and it does the same thing to the protein. And the longer it stretches the protein, the more the protein does something bad. It messes up developmental stuff. It'll kill you in your 30s and 40s and 50s. For a long time, there was a mystery of, okay, this is obviously being passed along, but it's getting worse, is that somebody who has Huntington's today is having a worse version of it than their father did, or their grandfather did, or their great-grandfather did. How's it possibly getting worse? And what these parasites do is they do lengthen themselves. They do break off a piece and put it somewhere else in the genome. But as they lengthen it, they mess with the code more and more. They take and they put more codons where there's not supposed to be codons and mess the protein up more and more and more, generation after generation. So, and there's a lot of these weird parasites that are literally in it only for themselves. Later, when we talk about genetics, we will come back and we will talk about what this, how this all works. Here's an article, fairly old article. 97% of the human genome that does not encode protein has taken a bad rap. But now this so-called junk DNA is turning out to play vital roles in normal genome function. A lot of processors and stuff there. So eventually people did wise up. They started to realize that the junk DNA isn't really junk. But took them a while. Okay, that's it for this particular chapter. We'll go on to cells, which are way bigger than molecules, next time.